Okay, this afternoon I'm joined by Dr. Mark Povroznik from WVU Medicine United Hospital Center. Dr. Povroznik is the Chief Quality Officer and Chairman of Infection Control at United Hospital Center. And we've asked him here to give us some information about treatments and immunizations that are available for the COVID-19 pandemic.
Well, thank you, Veronica, and welcome to the Immaculate Conception Parish Nurses. We spoke not too long ago, so for those that weren't on that call, it's a pleasure to be back speaking to you again. There really have been several treatments that have been part of the mainstay or growing in terms of therapy for COVID. And let's start by breaking them down in a few categories. Convalescent plasma, antivirals, and monoclonal antibodies. So to start with convalescent plasma, that literally means the plasma from recovered patients. That's the plasma portion that contains the antibodies that help to fight infection. And it's been used for more than 100 years to treat a variety of illnesses, from measles to polio to chickenpox and SARS. So it's a widely believed safe, effective modality. Now, initially, the researchers found that patients with or at risk of severe COVID-19 who received convalescent plasma within 3 days of diagnosis were less likely to die than patients that received convalescent plasma later in their illness. So the problem that came from this is that there was no control group to that study. So that's what we knew, and that's what's been a part of our therapy. But the question will remain as to whether or to what degree which convalescent plasma is effective for treating COVID-19, who would benefit from this treatment, and when in the course of illness is convalescent plasma most effective. And those are certainly questions that will come out as we continue in our journey of COVID-19, but that is what we know presently about convalescent plasma. Then we get into antivirals. The one in most widespread use is remdesivir, Veklury, if you're interested in the brand name. And what remdesivir is doing is binding to the viral RNA-dependent RNA polymerase. And by doing so, you're inhibiting viral replication and you're terminating that RNA transcription. So that's essentially what the drug is doing. And it can be used in those ages 12 and greater. And it may modestly speed up recovery time. But there are some caveats to who gets remdesivir. Patients first must be symptomatic. They must have worsening hypoxia requiring greater than or equal to 3 liters of oxygen. The therapy is to be initiated within 7 days of symptom onset, and the treatment duration is for 5 days only. And essentially, the exclusions would be those with liver dysfunction of AS or ALT greater than or equal 5 times the upper limit of normal, or patients on high flow oxygen therapy, ventilation, or ECMO. So that's kind of the caveat of where remdesivir is currently utilized. Now, what's making its debut and is coming out a little bit faster is monoclonal antibodies. And these fall into 2 categories. We have our man-made, and then we'll talk about one that was already on the market, which was a human monoclonal antibody that was used for different indications. But let's start with those that are man-made. They all have to have great long names that are tongue twisters to say, but it's bamlanivimab, and then there's a combination product, casirivimab, imendivimab. So let's just call it BAM and the combo drug. That'll be shorter. So we have 2 products, and they're man-made versions of antibodies that our bodies naturally make to fight invaders. They're designed to block the viral attachment and entry into human cells. So thus, it's neutralizing the virus and potentially preventing and treating COVID-19. Both are very similar in administration. Both are very similar in side effects. There are indications for administration. So initially, they're being made available to those greater than 65 years of age who have tested positive for COVID-19 within the past 8 to 10 days, not requiring oxygen or an increase in their oxygen baseline needs, not being hospitalized. This drug is intended for the outpatient setting, and those that have certain underlying chronic medical conditions. So it's really all of those steps, and you have underlying medical conditions such as a BMI, body mass index, greater than 35, chronic kidney disease, diabetes, immunosuppressive diseases, or currently receiving immunosuppressive treatment. So that's the initial rollout of who qualifies under the emergency use application to receive BAM or the combo drug. They are administered intravenously as a 1-hour infusion, and they require an additional hour of monitoring time. That's to assess basic parameters like blood pressure and heart rate, and ensure that you don't have a rare severe allergic reaction or anaphylaxis, which has only been one patient in each of the studies, so it's a rare occurrence. The benefits of treatment with monoclonal antibodies has not been observed in patients hospitalized due to COVID-19 and may be associated with worse clinical outcomes when given to hospitalized patients requiring high-flow oxygen or mechanical ventilation. So while it sounds like a great and novel therapy, those who receive it are to be outpatients with comorbidities and not requiring high-flow oxygen.
It sounds like that medication is almost meant to keep the COVID from escalating and getting to a more critical point. Is that correct?
That's exactly right. So by being a neutralizing antibody, and we can identify the higher risk patient types early on, then hopefully we prevent them from progressing to more severe illness. But once they do progress to more severe illness, this is not the mainstay of therapy for those patients. Now, there's one, go ahead.
No, just that was great. Thank you for clarifying that.
There is a man-made, or a human monoclonal antibody, Actemra, that was already on the market. It wasn't on the market for COVID-19, but this monoclonal antibody binds to interleukin-6 receptors. And there's some limited data from uncontrolled studies about the potential benefit for COVID-19, especially when they saw the severe cases that went more into a cytokine storm, and that cytokine storm patients had high levels of interleukin-6. So this is a therapy that is pulled out for those severe cases of cytokine storm in hospitalized patients called tocilizumab, or Actemra, that was there previously, or you might recognize it as a therapy for rheumatic disorders like rheumatoid arthritis. So first we had the convalescent plasma, then we had the antivirals, then the monoclonal antibodies. And, one of the questions I get when we talk about convalescent plasma is who can donate? So in order to donate plasma, a person has to achieve certain criteria. Obviously, they have had to have tested positive for COVID-19, because it's only after that you would potentially make antibodies. You have to have recovered, because certainly we don't want a sick patient going to donate plasma, and you have had to have no symptoms for 14 days. So you're beyond that infectious time period, you've recovered, you've had proof that you had the illness, and that you currently test negative for COVID-19. So that's part of the current criteria, but we also know that people continue to shed dead virus and can test positive for a while, it doesn't mean you're infectious, but these are the current donation rules. And you also have to have high enough antibody levels in their plasma to support donation, and that's something that donation facilities check for. And as well as a donor and a patient who receives it has to have compatible blood types. So once plasma is donated, obviously it's screened for other infectious illnesses like HIV, and if you're a donor that can give, your plasma will treat anywhere from 1 to 3 patients. So it can be a rich donation and support 3 people. And it's not thought to weaken your immune system in any way. And depending on the facility that you donate, you could donate again after 7 days. Those donation sites have their own rules, and it could be longer for some sites, but not shorter than 7.
Okay, great. That is a lot of great information for our community and our parishioners. I want to switch gears a little bit now and let you talk about the vaccines that are coming out in the very near future.
Certainly a hot topic, and one that I'm getting a lot of questions from our healthcare staff here at the hospital. So there are a variety of them there. So let's try to narrow down to the vaccines that are on the near horizon, and we'll start with those that perhaps are expected to ship in the next couple weeks. So if we were to put these into categories, vaccines are being developed through messenger RNA. They're being developed through vector technology, protein subunits, or whole inactivated virus. So we're going to start with messenger RNA. That's the Pfizer product or the Moderna product. Messenger RNA is really the newest technology, whereby they're using the genetic material called messenger RNA, and this is kind of like the playbook as how to make the coronavirus spike protein. So if you've seen the molecule of coronavirus on its outer shell, these little spikes, it's teaching your body how to make that little protein, so that protein then triggers your immune system to mount an immune response once you're exposed to COVID-19. And so they picked a piece of the virus that they thought would not mutate or change much. So we'll wait to see that in time, but the fact that both manufacturers are showing an efficacy rate around 95% currently means that it's not mutated during that process, and we hope that holds itself up. Now, the messenger RNA by itself is not very stable very long. So in order to package it up or to preserve it, it had to be wrapped into like a lipid or a fat molecule around the messenger RNA. And that then required low freezing temperatures in order to store it. So it's because of that we had to seek out these sub-zero freezers that could get down to negative 70 and 80 degree temperatures for storage. It's one therapy that's used during pandemics because it can be rapidly produced. That's probably why you see them coming to the market first. We don't need the virus itself when you're using messenger RNA. You don't need it at all. You just need the sequence of the messenger RNA that then tells your body how to make the spike protein. So these two vaccines will both require 2 doses, depending on the manufacturer, whether you get a second dose in 21 days or 28 days. Both have learned the same thing, that the initial dose is a small dose, followed by a larger dose for the second dose, which gets a higher efficacy rate than if you split the doses equally or did it vice versa. So that has been true for both products. And for the most part, that's the messenger RNA. And what's being told to the public and what we know in terms of side effects is what we would expect from a vaccine triggering our immune system. Muscle aches, headache, potentially for a low-grade fever, a sore arm. The way I like to tell people, well, what's the difference from the flu? Those that have had the flu shot say, well, I get nothing or I get mild symptoms. Those that go on to, in their later years, to get, say, the varicella vaccine or the shingles vaccine, say, oh, man, my arm really hurt. Yes, you got a vaccine that you weren't used to, never exposed to, and you got more of a symptomatology from it. So that's how you should think about this vaccine. You've not had it before. It's not unexpected that you're going to get a sore arm or feel a little fatigue for a couple days, which is why the recommendation is for any group of patients or employees that we don't vaccinate more than 20% of them on any given day, that we kind of stagger those vaccines for the same work group. So we're going to anticipate you have some of those side effects.
Yeah, and I think that's an important point to make for community members, is that you're not, it's not a live vaccine, if I understand what you're telling me correctly. So they're not getting the virus, they're just getting something that's going to make their body recognize the virus. And then also that they can expect to feel bad a little bit, and that doesn't mean that they've contracted the virus from the immunization, I think, which is a common misconception.
It's a great misconception. It happens every year that we do the flu shots. There are those that just have geared up immune systems, and they say, boy, I got some aches and pains, and I got the flu from this. No, you didn't. Your immune system reacted, and that's a great thing. And it's important to talk about, to expect it, because you need to go back and get your second vaccine and not run away from it. So the first shot is where it's expected to have some of this discomfort. The second shot, even though it's a larger dose, most are reporting little to no symptoms from that shot. Now, also on the horizon are vector vaccines. So some people recognize company names. So that's AstraZeneca's product. Vector vaccine uses another virus to carry the genetic instructions into us, so that we then make that same spike protein. So one is using messenger RNA to give us the roadmap to make it. A vector vaccine is using a different virus as the carrier to give us that roadmap to make it. And that carrier is called adenovirus. And there are different forms of adenovirus. The one that AstraZeneca is utilizing is one that infects chimpanzees. It doesn't infect us, it infects them. So they took that virus and they modified it so it doesn't cause disease, allowed it to be the carrier in terms of vaccine distribution. And in the end, we make the same spike protein so our immune system recognizes it and is able to mount an immune response quicker once if we're exposed to COVID-19. It's a cheaper way to make vaccines. So cheaper doesn't mean less effective. It's a cheaper way to do it. And as you can imagine, trying to freeze something down to negative 70 degrees is an expensive process. So this process is a cheaper process, but it's slower than using messenger RNA process. But once it comes to market, it could be an opportunity for a vaccine to be more stable at refrigerator-type temperatures for up to a couple months, as opposed to the messenger RNAs have a short half-life once they're, or short stability, I should say, once they're taken and thawed out of the freezer. They too will likely be 2 doses, but just a different delivery system. And right on the heels of a vector vaccine delivery system is one by Johnson & Johnson. And here's where they also use an adenovirus, but it's not the chimpanzee. It is the human adenovirus 26, which can cause the common cold, but they've disabled it from doing so. But similarly, they're using a virus as the carrier to enable our immune systems to make that spike protein. And that's the delivery system that was used back during the Ebola pandemic, using adenovirus as the carrier. Then you can get into what's called a protein subunit. And this is a company like Sanofi, Novavax, that's working on this technology. They take subunits, little pieces of the target virus, and that's what's utilized to mount an immune response. So instead of using the human body as the vaccine factory, which is what we did with a vector vaccine, with a protein subunit vaccine, they're actually using an insect virus called baculovirus to help produce these protein molecules. And they are done, okay, wait for this one, they are done inside of a moth. So if you've been reading about this, this isn't science fiction, this is how it's done. So the insect virus is in moths. They use that to make the proteins. We harvest the proteins out of the moth, then to make the vaccine. So sounds like science fiction, but it's not. It's been around for a while. Hepatitis B vaccine used commonly in newborns, as well as the human papillomavirus or HPV vaccine is made the same way. So I'm just bringing out these little subtle differences so that when you read about them, you think, well, okay, this is a reputable site that I'm reading about. This is the way it's done. And then lastly, you would have a whole inactivated virus. So this is what a Chinese company called Sinovac has done. You actually grow coronavirus, and then you kill it by chemicals or heat, and then it's that inactivated virus that's used to make a vaccine. So those are the four ways. The one that's expected to be shipped at any time is the Pfizer messenger RNA vaccine, and that's also the one that was approved yesterday in the United Kingdom.
Great. It sounds like we have some very promising vaccines on their way. One thing I want to back up, you talked about the 2 doses, and I think it's important to point out to people that they won't get full immunization from just that first dose, and that they need to get that second dose. And then once they get that second dose, when would they expect to have to be more safe? Because we still need to follow the precautions for a while. We can't assume that we're getting rid of masks and social distancing tomorrow. So what would you say to people about when they will be fully?
Yeah, a lot of details are still unknown, and healthcare as a whole is still looking for some of these details. So, certainly once the FDA meets and releases, more information will come. In general, when you administer vaccines, the effectiveness of that vaccine is not for 14 days after. And the CDC is clearly telling us that for now, the vaccine does not preclude our existing safety measures. So those that get it should not say, well, I don't have to wear a mask because I got the vaccine. That is not true. So we need mass vaccination. We need to then continue to monitor to say, okay, if you're exposed, do you shed it? If you're exposed, do you get it? And until those answers are known, our continued safety practices will stay in place, and we'll still do our masking.
Great. Thank you for clarifying on that. Now, you talked about a lot of great stuff. The last things that I think we want to talk about is the rollout of this. And I know there's been no definitive plan laid out, but I think there are some rumblings out there. So what are you thinking about the rollout of these vaccines?
Well, what we're being told when they're released, they'll go to state distribution centers. And one of those distribution centers will be Berkeley in the Panhandle, and in Central West Virginia, it'll be Ruby Memorial. And from that central distribution center, it will go out to the other hospitals that have the capability to store the product. Again, it's got to be kept very, very cold. The initial rollout is in tiers, starting with direct healthcare providers caring for COVID patients. So hospitals will follow the algorithm that has been handed to us by the state, and it's not surprising that initial tier level is your emergency room doctors and nurses, your critical care nurses and doctors, those that are operating a COVID unit. And that's the first group. And then from there, it'll quickly spread out to care providers that are involved in the care of those kind of patients, physical therapists and lab and phlebotomy and anesthesia, and it'll quickly go. But the top point was the ED, critical care, and COVID units. So those vaccines have been ordered, and that's what we expect to receive. And the state has been clear that the process has to be to log these vaccines into a VAMS software system, vaccine administration medical or system of some sort, where they will keep track. And based on those allocations, you will receive equal number of vaccines to those that received it the first time. So they will earmark and hold back those vaccines for those that need their second dose. So that's the initial rollout, is those highly targeted people, high-risk individuals. The word is that, they want some fair distribution to the greater than 65 with comorbidities. How that's going to roll out through the state, I do not have a lot of information on yet. That's the intent. And I think the real impetus is for the FDA and Pfizer to sit down and get the approval process going, start the distribution, start the recording of vaccines so that we can roll out as quickly as possible.
Great. This has been a wonderful, very informative interview session. Is there anything else that you would like to share before we wrap up?
Not that I can think of at this point, other than, if you're going to research information, do reputable sites, go to the CDC websites. The start of vaccine production really started with a lot of negative publicity by people that really didn't know. So stick to the facts of what we do know. Messenger RNA is not a live virus. It's just a template. We're training our bodies to make a protein so that we can recognize it quicker. That, yes, we should expect to have some side effects because it's new, just as if you get new vaccines in your later years of life. And this is the first step to help us get through the pandemic, and we'll learn as we go what kind of immunity we get from it, how long it lasts, does it stop virus shedding, and create some sense of, hopefully, normalcy in the near future.
Yeah, and I think that's the end result we would all like to see. Well, thank you, Mark, for this very informative session this evening. And you're always a friend to our parish nurses, and we appreciate you so much.
Well, I appreciate you.